it's great to be here, and as David said, this is actually quite a unique gathering, a small group, people coming at a common problem from very different angles, and I think this is a great way to kind of come up with new ideas as we have time to mingle and talk to each other also. So the title, I can't remember if I gave this title originally or if it was given to me, was supposed to be Central Mechanisms of Pain and fMRI Analysis, but as I was putting my talk together, my lab does a lot more than just fMRI, so I've thrown in other stuff too, so I hope that's okay to fill things out. Okay, so I'm going to start with this simple model of Descartes, which I'm sure everyone is quite familiar with, because it's a good starting point to think about historically. The model of how we feel pain was extremely simplistic. You know, you stick your foot in the fire, and a bell rings off someplace in the brain, and boom, you feel pain. Well, of course, we know it's a lot more complex than that, and we know that there's a lot of different dimensions of pain. And what became very curious to those of us in my lab over the last few years is this issue of nature versus nurture. So we've been studying individual differences in how people experience different types of pain, acute pain in healthy individuals, but also why is it that some people develop chronic pain and other people don't develop chronic pain from the same injury. So we chatted a little bit about this last night at Linda's talk about what makes some people resilient and other people more vulnerable to develop these sorts of things. So with that kind of thinking in mind, we've been trying to come up with ways to kind of see what the different contributions are, because down the road, wouldn't it be great if we can all be the resilient type? If we were given a set of genes or connections that made us vulnerable to something, how can we become more resilient? So I'm going to very briefly at some point in my talk a little bit about personality, because it's one possible way of looking at vulnerabilities. Certain types of personality attributes are considered to be relatively stable over time, and they may give us a little bit of insight into how we're pre-wired and perhaps leave us vulnerable or resilient to develop chronic pain. The flip side of that is, how does our brain change, and what is the plasticity that's associated with prolonged nociceptive input? So how does that change the brain? So that's more of a kind of the nurture thing. So we're looking at these two aspects. And the third area that we've been looking at in my lab is how pain and attention interact with each other, not only to enhance each other, but also problems in cognition and attention when you have pain, and vice versa. And can we perhaps even use or harness kind of attentional or cognitive mechanisms to help people cope with pain better? So that's the basic concept of the kinds of studies that we've been doing in the lab to get at these sorts of issues. So neuroimaging, why are we doing this? Is it just a basic tool to look at how people are wired and what their response is in the brain, or can we actually eventually move to clinical relevance? And it's nice to follow David, because he set up a whole host of possible clinical applications of neuroimaging. So I think that's great. And I'm just going to touch upon a few by setting up some questions now, and then we'll return to these questions after I show you some data and some of the technologies. So perhaps neuroimaging can be used to determine when is the best time to intervene with a treatment. Perhaps neuroimaging can be used to look at potential side effects of treatment. So that includes things like what David was talking about in drug development. Can we use neuroimaging to give us insight into what might be the side effects of this drug or this other type of treatment? Thirdly, how do treatments like deep brain stimulation and others affect the brain? And finally, what is really, again, the main focus of my lab is to understand individual differences and how those individual factors may impact treatment outcome. Okay, so let me just give you a little bit of an update about the developments of neuroimaging that are hopefully moving us towards using imaging for these clinical applications. First is a way of doing functional MRI called Percept-related fMRI, and I'll describe that in a few minutes. Secondly, David talked about resting state functional MRI or functional connectivity. Third is the advent of structural MRI, being able to image and measure gray and white matter. And finally is putting imaging of pain, plasticity, and tension together into a context where we're able to look at the contribution of these individual or pre-existing vulnerabilities versus disease-driven factors. Okay, so this is kind of an overview of how we approach imaging in my lab. I guess the trackball here. Oh, here it's working. So this is the main way that people do functional MRI. And maybe I should just take a show of hands who here actually does functional MRI. Okay, so just a small number. Okay, so I'll go through this a little bit just so everybody understands this. I'm actually going to spend a fair bit of time talking about the technologies so that you can understand other speakers when they show imaging results. So basically, how fMRI is often done is that a stimulus is delivered, so let's say a noxious stimulus applied to the skin, in what we call a boxcar fashion. Kind of a certain amount of time you're delivering a stimulus, and then you stop for a certain amount of time and you repeat that. And what you do then is you take this time course waveform, this up and down waveform, and you do what's called a mathematical convolution, kind of like a multiplication, but not quite, with a waveform called a hemodynamic response function that looks something like this. And what that does is it takes the time course of the stimulus waveform that you're delivering, and it puts it into a time frame of what the hemodynamics are expected to do in the brain. So you have to keep in mind that when we image the brain with fMRI, we're looking at what's called a BOLD response, which is the blood oxygen level detection response, which is a hemodynamic response. It's very, very slow. Coming from an electrophysiology background, it also doesn't feel quite right because we're talking about things that are happening on a time frame of seconds, many seconds, as opposed to milliseconds. But that's the course time frame that we're looking at, changes in the hemodynamics in the brain. So then what you can do is you can look in the brain and you find hemodynamic changes in the brain that match this basic waveform that you've constructed. And you can color code those responses through after doing some statistics. And that's basically your brain response to the stimulus. And you can then manipulate attention, you can look at the effect of personality, you can look at brain structure and connectivity, and you can do all those fancy things. Okay, so this is a lovely diagram that I stole from one of David's reviews. David produces fantastic images in all his reviews that I constantly steal, so thank you. So this explains it in a little bit more detail, that here's your stimulus waveform, on, off, on, off, on, off. This could be a painful stimulus applied to the hand, let's say, and this is your expected, or actually recorded fMRI signal that you found in this area of the brain because it matches this waveform once it gets convolved with the hemodynamic waveform. So you're looking for this in the brain, and when you do the statistics of whether or not there's a statistical difference between this level here and a baseline level, you color code those, and that's basically what you're doing when you do task-evoked fMRI. Okay. So that's great, and there's been lots and lots of studies out there, not just in the pain field and across all fields, that use that approach, and it works just fine. The problem, what started to really bother me when we first started to do fMRI in the lab, is that we recognize that pain is different than applying an auditory stimulus or a visual stimulus or a touch stimulus or something like that. And the reason it's different is because the perceptual response evoked by a maintained stimulus, the time course of that isn't always a one-to-one response. Sometimes you're feeling more and more pain as you maintain a stimulus. You might habituate to that stimulus after a certain period of time, or you might not feel anything until the stimulus is on for a certain period of time. So there's many temporal dynamics issues that indicate that the evoked perception doesn't necessarily match the stimulus. So if you're looking in the brain for this hemodynamic response, that can certainly tell you about a stimulus-evoked activation, but it doesn't always tell you about specific types of pain and where the representation is in the brain of that kind of pain. So, so what we did, and some others have done the same thing, like Vanya Apkarian and Carlo Porro, is we developed something called Percept-related fMRI, and this was about 10 years ago. And what Percept-related fMRI is that, it's hard to see here, but see this little squiggle here is a continuous rating of a sensation, let's say intense pain intensity or something like that, that you ask the subject to provide while they're being imaged. So this is online, continuous rating of anything you like, any perception you like. You tell them what to rate, and they use a little trackball system, whatever system you develop. And then you take, instead of taking this waveform of when the stimulus is applied, you take this waveform of the actual time course of the perception, you convolve that with the hemodynamic response, and now you've got a new waveform that you're looking in the brain for a signature of, and that gives you an index of the brain activity related to specifically to that percept. Okay, so so it's a slight nuance, but it's important for imaging pain, I think. So I'll give you some examples of how we first applied this. This was about 10 years ago. Our first system to look at this, to see how it worked, was a system where we tried to evoke different qualities of pain, and we used the model system of cold. And the reason we use this system is because when you deliver a very cold stimulus to the skin that produces pain, it produces many other sensations that tend to have very different time courses. And they have different time courses from person to person. There's lots of individual variation. And we can take advantage of that. So we first looked at prickle. We've actually looked at a lot of qualities of pain. And again, the qualities of pain, it wasn't just to look at this percept-related trick, but it's also because different qualities of pain I think give us different insight into clinical conditions, because we know that diabetic neuropathy pain feels very different than post-stroke pain, etc. So each of those qualities that we're experiencing must be represented somewhere in the brain, and if we can distinguish one from another, maybe we'll have a better target for therapy down the road. So here we can cool the skin down to about 0 degrees. The person can feel cold through all that, but they occasionally start to feel these sharp prickles. Of course, I come from Canada, so we're quite familiar with this in the winter time. Now, this box shows individual ratings. The blue bars are the cold stimulus, and each green line are the individual ratings of individual subjects of prickle. So we ask them to rate the prickle when they feel the prickle. And you can see that it's all over the map, right? Everybody's feeling things at a different time course, and as a psychophysical experiment, it's a mess. But for percept-related fMRI, it's fantastic, because we can say now, show me where in the brain, when we convolve each of these blips with the hemodynamic response, show me where in the brain there's an activation each and every time. Okay, so we do a very high-level statistical analysis to find that, and we were able to find a set of brain areas that were uniquely activated to the prickle sensation. Okay, and I won't go into all the details, but they represent systems involved in attention and in pain and in something moving across the skin and all sorts of things like that. We used the same kind of model to then look at another percept that is detected when you also cool the skin in the same experiment, but now we ask the person in another set of runs, tell me when you're feeling paradoxically a burning sensation, a heat sensation. And again, for us Canadians, when we come inside after being outside for a very long time, we take off our gloves, and you often get this paradoxical heat sensation. And the interesting thing is, if you measure the temperature on the skin, your hands in this context may not be hot at all. They may be a neutral temperature, but you're feeling paradoxical heat. Again, this has some clinical relevance. And so when we do the experiment, again, ask people to rate a heat sensation and do the Percept-related fMRI, we actually only got one area of the brain that was activated, and that was included the area around the right anterior insula. And around that time, this was in the early 2000s, was when Bud Craig and others were talking about this right anterior insula having to do with many aspects of thermal sensation and introspection and attention. So it seemed to fit. Okay, so how would we then use this approach to look at chronic pain? Well, just like an evoked paradoxical heat or evoked prickle might occur at a different time related to when the stimulus is applied, chronic pain also can deviate from when you deliver a stimulus. So I'm going to show you this data, which is derived. Sorry, this trackball is not working well, so maybe I'll go back to this. Okay, so this is derived from experiments that we did throughout the 2000s in patients with irritable bowel syndrome. And what we did in these experiments is we used a paradigm of rectal distension to both look at the physiology and the psychophysics involved in rectal distension in the gut, and then also look at the brain response. So in a control subject, the black line indicates the pressure of the inflated balloon in the rectum, and the thick line indicates when subjects are rating the intensity of pain. And in normal control, healthy subjects, basically when the balloon is inflated, they feel pain, and when it's deflated, the pain goes away and everything is fine. The curve is a little bit shifted, and that's just a time delay due to the inflation of the balloon. In irritable bowel patients, there's a total disconnect between the time course of the stimulus, the inflation of the rectal balloon, and when they're feeling pain. So they start, this is just one subject, an example, so they may start to feel pain, but when the balloon distension is removed, their pain remains up. And you send it again, and the pain gets even worse, and it may go down, but as you can see, it's a disconnect. So this is where this approach of using Percept-related fMRI became quite important. If we wanted to look at what happens in the brain when they're feeling pain, then we absolutely have to know when they're feeling pain. So we use this Percept-related response. We had them rating what they're feeling in the scanner as we're collecting data, do the mathematical convolution, and then we create what's called a new predictor curve. We look for that time course in the brain, and then we're able to identify pain-related activations. Now, of course, we can still take the time course of the stimulus, of the rectal pressure stimulus, and go through the regular fMRI approach, and then we would be identifying stimulus-related activations, and those actually are quite important too, because they're telling us when the stimulus is on, what is happening in the brain. The important thing is that these two approaches give you different types of information that can come together to tell you what's wrong with that clinical condition. So I won't go through all the data of all those experiments, but just some highlights when we did these experiments is that we're actually quite surprised. We thought, okay, people are feeling more pain when they have irritable bowel syndrome. We're going to get all this additional activation, and we did get greater activation in some pain areas. But we were at first perplexed that we actually had little or no activation in the patients compared to controls in two areas, one being the insula and the other being the anterior cingulate cortex here. And we didn't know what that meant at the time. We kind of thought, well, maybe there's a problem with triggering proper interoception and homeostasis. So this brain-gut status update's not working well. We're not triggering the descending controls, etc. We didn't know, but we were still feeling a little odd about this finding. What should we do? So hold that in your memory for now, because I'm going to come back to the follow-up study that we did a few years later. But before I get back to that, I want to finish describing to you the approaches of different types of functional MRI. So we talked about these evoked responses when you deliver a stimulus, but another approach that is a newer game to play that David spoke of is functional connectivity. So functional connectivity is an interesting term, but it doesn't actually mean that two areas of the brain have a synapse between a neuron here and a neuron here. It does not mean that. It simply means that the overall activity in this area of the brain and this area of the brain are correlated. So they're doing similar things. So the term was introduced of functional connectivity to describe that they're doing something similar. They may be getting a common input, or they may in fact be speaking to each other, or they may be just having a similar activity profile that are doing two things that happen to have the same temporal dynamics. We don't know, but that's what a lot of people are looking at, and it can give us some very interesting insights. So what are we using this approach for? Well, one approach is to look at networks. Okay, so we know that pain is not just due to one area of the brain, but there's a network response to many things. So people looking at functionally connected networks are using this approach. And basically, so Friston and others first described this idea of correlations between spatially remote areas, and we can use very complex statistical analyses to pull these areas out. What was first described, the first really network that was described, is called the default mode. And this is, some people don't like that term, but basically it's what's happening in the brain when you don't deliver a task or have any sort of stimulus. And so it was first described by Reichel in 2001, and there was a series of areas, precuneus, posterior cingulate, medial prefrontal cortex, and some others, that showed a decreased activity during a task. So they called it a task negative. So it seems to be that these areas are kind of on when you're not doing anything, and then they get suppressed when you have to do something to allow that something to occur. Now, the interesting thing about these networks is they're operating and they're identified by looking at very, very low frequencies, less than 0.1 hertz. Okay, so this is where I really struggle to try to understand what the meaning of these are, because this is really slow, right? Regardless, they're very, very robust, and you can find them all the time. So maybe Reichel really talked about these states as being important for maintaining some sort of monitoring of your internal environment and your external environment, so that you can react to something that then comes along and focus it. Sorry? Oh, okay. I'm going to move along faster. So we've identified many, and others have identified many of these intrinsic networks, and that might be involved in a variety of different states. Another approach, though, of this functional connectivity is a more focused approach, looking at just how one area talks to another area. So you pull out the activity of one area called the seed, and you say, okay, how do other areas correlate with that? And basically, we've done that to look at how different parts of the insula talk to different parts of the cingulate cortex, and have found various patterns of connectivity. And so now that we have this framework, we can look at what happens in a disease or pain state. So let me just mention now structural MRI, because that's a great tool we have in our arsenal, kind of the highways and cities of our brain. Now, structural MRI has two components. You can look at gray matter, and you can measure cortical thickness or subcortical volumes. And the problem is we don't know, when we do these measurements and we see changes in pain patients, we don't know what factors of all these possible factors contribute to that. So those are still being developed. There's a lot of animal studies being done to look at what factors might change to give us a different readout. But nonetheless, we and others continue to find altered structural properties in pain patients. So this is an example from the IBS patients. So we showed functional MRI that there was no response or little response in the insula and cingulate. When we went back and we reanalyzed that data structurally, but we also collected a second group of data, we found indeed there was cortical thinning in those areas that showed no fMRI response. So maybe they showed no fMRI response because there were fewer neurons or systems there to mount a response. So that seems to make sense. And we also interestingly found an enlarged hypothalamus, which might speak to the stress response. You can also look at things like the impact of pain duration over time, and we see that there's a difference in cortical thickness depending on how long somebody's had IBS, and so that speaks to long-term persistent pain and plasticity. I won't mention this too much because we're still doing these studies in temporal mandibular disorder, but we're finding similar things in temporal mandibular disorder that you have changes in gray matter that also may change with time, but also change depending on how intense the pain is. So again, talking about persistent input to the brain over time changes the brain. So is it nature or nurture? And maybe it's a bit of both, because we do have some impact of personality on this, but we also have impact of time and pain intensity. The news is not all bad. The good news is that there are a series of studies that have come out in the last 2-3 years showing that many of these brain abnormalities and chronic pain can be totally reversed back to a normal state after you get rid of the pain. And this is an example from a group looking at before and after hip replacement surgery. What about white matter? So David talked about white matter and using diffusion tensor imaging, so basically looking at water diffusion. Interestingly, some of the markers that we can measure using diffusion tensor imaging are markers of neuroinflammation. Okay, so the radial diffusivity and mean diffusivity show signs of neuroinflammation. So and there's other factors as well. So we're now looking at white matter changes in a different light because of this. Although most people measure FA, we can also measure these other markers of neuroinflammation. And we can also create beautiful pictures using all different types of approaches. And I'll just flip through these, but you can actually visualize peripheral nerves as well as white matter tracts in the brain. And indeed, one example of looking at white matter in the brain of IBS patients showed that the fornix showed an increased FA, perhaps due to this limbic system or HPA response. So in about 2 more minutes, I'm just going to give you one other set of experiments I think is important for trying to understand how we might be able to manipulate pain or the vulnerabilities or resilience of patients with pain based on how they balance their attention and capacities. So this was a study done by Dave Siminowitz in my lab a number of years ago when he was my Ph.D. student, and he did it. He gave people, normal, healthy people, a task called the counting Stroop, and with and without having an acute pain stimulus delivered. And there were 2 types of responses that we found that we were surprised. We assumed that everybody, once you delivered pain while they were doing the task, you would assume it would interfere with them doing the task and they would get slower. But half the group's subjects actually got better at doing the task, and they told us that they concentrated more and they tried to not think about the pain while they were trying to really do well in the task. And when we imaged their brains while they were doing this, these people, we called them attention people versus the pain people, these people modulated their brain. This is sensory cortex. They modulated their brain while they were trying to do the task with the pain on board. So the brain was doing and reflecting the behavior. So it could be that there's different coping strategies, but obviously different wiring and organization of the brain in these 2 types. And if we can harness that for modulating their responses, that this might help their pain. So the question is, how are these 2 groups of people wired differently exactly? And those are the sorts of things we're looking at now. Certainly we know, this is back to pain patients, that the pain patients do show an abnormal behavioral response to cognitive tasks like the Stroop, and we do know that those kinds of patients, this is again back in TMD, show a different response in the brain to those cognitive tasks that show changes due to cognitive interference and emotional interference, and that the coupling between different areas in the brain is off for those patients. So there's certainly an issue of behavior and brain structure and function, and I don't have time to go through this all, but we certainly have an impact of things like pain catastrophizing and neuroticism on how the brain is organized. So I don't think I have time to go through this because we're running short. But just to mention that both the pain patients and normal controls show a different organization, both in terms of functional and structural organization in the brain, depending on their personality profile. So I won't go through this. So basically what we're saying is the individuality gives you your vulnerabilities and your resilience, and we need to harness that so that we can try to find a way to maybe move towards different types of personalized treatment. So just in closing then, starting with those questions that I talked about earlier, what's the clinical relevance of imaging? We can choose the best time for intervention or treatment if we know the time course of this disease-driven brain plasticity. We might have a better idea of when to intervene. By looking at connectivity, we might be able to understand better the potential side effects, because we know if you treat this area, if it's connected to all these areas, what side effects might occur. We can evaluate the effect of treatments on the brain by using now a variety of metrics of plasticity, not just fMRI, but all these connectivity, functional and structural connectivity measures. And finally, I think one of the most exciting things that I see coming out in the future of personalized medicine is to understand individual factors that leave people either resilient or vulnerable to these things. So brain imaging has a great potential for the future. I just want to, not to leave you with a downer, but just to leave you with a caution that we need to really be careful about issues surrounding neuroethics of brain imaging in people in terms of the doing good imaging, so that we know that the data that we're getting are good data. And certainly anybody at this meeting is doing good imaging, but there are people out there that are doing not good imaging. So we have to be very, very careful. So thanks very much.
So we have time for one very quick question.
Sorry. I didn't get a chance to ask this question for the previous presentation, but it applies also to this presentation. I'm not sure if the pediatric subjects in the study that was mentioned by Dr. Borsuk were under anesthesia, but how would you approach this, especially when you're trying to reconcile the human data with the animal data? And is this a problem really interpreting a truly painful stimulus?
No, absolutely. Well, so in my presentation, nobody's under anesthesia, but certainly that's a problem. And it's a problem for more than one reason. If you're doing fMRI, there's issues about hemodynamics that you have to worry about if you may have changed the hemodynamic capability by being under any drug, actually, or even people with stroke or anything. So you have to think about those technical aspects. Certainly, if you're under anesthesia, you're not feeling pain. There's no pain. So you can't do fMRI of pain. You can do fMRI of nociception, but not of pain, right? So you have to understand that distinction. Certainly at somewhere in the brain, something is happening to put you under anesthesia, right? And whether or not it's the coupling between areas that is falling apart when you're under anesthesia, or there's certain brain areas that do shut down, those things are really still not known. But definitely the animal studies in an anesthetized state can't directly be compared to the human. We have to consider those things. It's a problem.